Thanks for taking time to view this on-demand recording for Poster 978 as part of the Controlled Release Society's virtual annual meeting and exposition. The topic is a clinical assessment of delayed release coated capsule compositions for regional gastrointestinal delivery using gamma scintigraphy. I'm Chris Rowe and I'm a Senior Research Fellow in Scientific Consulting at Quotient Sciences in Nottingham in the UK. And I'm presenting on behalf of my co-authors Beth Brickhill, Litza McKenzie, Simon Yao and John McDermott at Quotient, and on behalf of the study sponsors, Steve King of RTS Bioconsulting and Derek McLean of Applied Molecular Transport. In terms of learning objectives, I hope this presentation can help with understanding the utility of gamma scintigraphy in both tracking the movement of oral dosages through the GI tract and in assessing the anatomical location of disintegration and release of contents from different delayed release coated capsules. In addition, I'll share information regarding the impact of fed versus fasted dosing on the GI transit of a selected coated capsule system. The rationale for the study is that Applied Molecular Transport are evaluating targeted drug delivery capsule systems to administer drugs with local activities to specific sites in the GI tract. Gamma scintigraphy was used to evaluate GI transit and release from 3 different coated capsule prototypes. In addition, pH, temperature, and pressure data were measured from a co-administered smart pill with particular capsule doses. The study enrolled 8 male and 4 female healthy volunteers who met the inclusion criteria, as were all non-smokers with body mass index between 20.5 to 30.1 kilograms per meter squared and no history of GI disease or surgery. These subjects received 3 delayed release coated size 0 capsules containing the radio label in a fasted state. Following a within-study interim review of emerging data, the subjects subsequently received one of the 3 coated capsules again in the fed state. Figure 1 on the right of this slide is a composite of the gamma camera data generated for an individual study subject and comprises an overlay of selected scintigraphy data collected at 0.25, 4.75, 6.75, and 8 hours post-dose. The white outlines of the stomach and colon for this subject were generated within the scintigraphy analysis software based on a 99M technetium radio-labeled drink taken by each subject alongside dosing of the indium radio-labeled test capsule. The colored pixels visible represent the actual amount of radiation reaching the gamma camera face, and you can see that the capsule with its radio-labeled contents remain intact through the 0.25 hour time point where it's in the stomach, and at 4.75 hours where it's actually in the small intestine, which isn't outlined in this composite. Initial release of radiolabel is observed as a halo of radiation around the remaining radiolabeled core of the capsule in the image at 6.75 hours, after it has entered the ascending colon. Finally, we see complete release of radioactive contents of the capsule in this subject at the 8-hour time point, still within the ascending colon, where a dispersed cloud of radiation has been imaged. The capsules dosed in the study were radio-labeled by drilling a small hole in the coated capsule shell, filling with a short half-life radio-labeled indium-111, before sealing the hole with a surgical bone cement. A pre-dose formulation development package was completed to ensure there were no significant impact on the test product coating integrity or dissolution performance. The 3 test capsule coating systems comprised of different ratios and coating weight gains for mixtures of Erdogat FS30D and L30D55 polymers, which dissolve above pH 7 and pH 5.5 respectively. Coating 1 contained a 50-50 mix of the 2 polymers and a 75 mg per capsule coating weight gain without capsule banding. Coating 2 had a 70-30 mix of the pH 7 to pH 5.5 dissolving polymers, also at a 75 mg weight gain, but this time with capsule banding, and coating 3 was an 80-20 mix of the pH 7 and pH 5.5 dissolving polymers at a lower weight gain of 45 milligrams, but also with capsule banding. As mentioned earlier, we also took the opportunity to generate a single reference pH, temperature, and pressure versus time profile for each volunteer subject in the study by co-administering a smart pill in a different set of 4 subjects during each of the 3 initial fasted periods of dosing. And this allowed us to compare transit times from the smart pill against the radio-labeled coated capsules on at least one occasion for each subject. The fasted scintigraphy results for all 3 coated capsules are shown here in the slide. The bar charts on the right have GI locations of release on the horizontal axis and the number of subjects on the vertical axis. Figure 4 shows the gamma scintigraphy derived locations for initial release, whilst Figure 5 shows complete radiolabeled release, where there's no longer a core of radiation visible in the gamma camera images. Some GI locations are joint, where the radiation is observed across both locations in that specific image. Capsule coating 1 gave the earliest mean initial and complete release times at 2.7 hours and 3.8 hours respectively, and the green bars in the chart show the early and relatively wide spread of GI release locations for this formulation. Capsule coating 2 gave the slowest mean times for initial and complete release at 4.9 hours and 6.2 hours respectively, and in blue on the charts, all the release was located between the distal small bowel and the ascending colon. Capsule coating 3 gave mean times between those of the other two formulations, and the yellow bars show a larger range of release locations in comparison to coating 2. Following a review of this scintigraphy data from the fasted dosing, it was decided to select capsule coating 2 to administer again in the fed state. On slide 6, we have similar bar charts in figures 6 and 7, showing the same fasted data for capsule coating 2 in blue, but this time with the fed data for the coating system now added in orange for comparison. In the fed state, as could be anticipated, the mean initial and complete disintegration times were delayed by around 10 and 9 hours respectively. Although for 7 subjects where GI location of release could be observed, the locations were the same whether fed or fasted, indicating that the pH-based dissolution of the coating system was performing robustly and worked despite prolonged GI transit times in the fed state. Gamma scintigraphy in this study was up to 24 hours, and at that point, 4 subjects in the fed state didn't yet have capsule release due to the extremely elongated GI transit times. And as such, any gamma scintigraphy monitoring of fed dosing with this formulation system in future would be recommended to continue to around 48 hours post-dose. On a volunteer safety note, only 3 mild adverse events were reported by our principal investigator during the study, and none of these were related to the coated test capsules or the smart pill. Figure 8 on this slide shows a typical example of smart pill data we generated, in this case for subject 001. The red profile is pressure, blue is temperature, and green is pH data, all against time on the horizontal axis. The thick grey and green vertical lines are the key times of gastric emptying and colon arrival. It can be seen that pH rises rapidly from around 1 to 7 upon gastric emptying, whilst there's a typical drop in pH coincidental with a pressure profile change at the point of colon arrival. The overall smart pill GI transit times, pressure, temperatures and pHs were found to be as would be expected for a healthy volunteer population in the fasted state. Where smart pill data could be compared to scintigraphy results from the same dosing occasion, we've got the data here in table 1. The gastric emptying times indicated that the test products and the smart pill didn't transit at a similar rate in all cases, perhaps due to size and surface material differences. However, the pHs measured by the smart pill at the locations where initial capsule disintegration was identified by scintigraphy indicates that the coating systems were dissolving at pHs between 6 and 7, which was in line with expectations for the polymers we used. So in conclusion, this study successfully compared the in vivo performance of 3 different capsule coatings using gamma scintigraphy. There were no significant safety issues during the study, and the results identified a coating formulation for future development with acceptable variability and the potential to deliver to a specific area of the GI tract. I'd like to thank you for watching this recording, and I've included my email address here should anyone wish to reach out for further information about the study or gamma scintigraphy in general. Thank you.